When blood glucose levels are high, as they tend to be during absorption of a meal, insulin is released by the pancreas. Increased insulin triggers several metabolic adaptations to increase nutrient absorption. Insulin stimulates the transport of glucose into body cells, where it is used for ATP generation via glucose catabolism. Insulin stimulates the conversion of glucose into glycogen for storage in liver and muscle cells. Insulin stimulates the synthesis of triglycerides in liver and fat cells. Insulin stimulates the use of amino acids for the production of new proteins. As you can see, insulin is the primary hormone regulating metabolism during the absorptive state. The net results of these metabolic activities are providing glucose for oxidation and generation of ATP, storage of fuel molecules in liver, muscles, and fat cells, and providing amino acids for the production of proteins. The major metabolic challenge in the post-absorptive state is to sustain a normal blood glucose level. When blood glucose levels drop, as they do several hours after a meal, then the hormones glucagon and cortisol are released. Under low blood glucose, the autonomic nervous system is also stimulated to release epinephrine and norepinephrine. After fasting for a few hours, epinephrine and glucagon stimulate the breakdown of glycogen in liver cells, thereby increasing available blood glucose. Fasting for many hours or a few days depletes glycogen reserves, causing the breakdown of proteins and lipids to become a major source for energy. Cortisol and glucagon both stimulate metabolic reactions that cause liver cells to produce new glucose from lactic and amino acids. The breakdown of lipids, stimulated by epinephrine and norepinephrine, also results in increased glucose production. If fasting continues, then fuel sources other than glucose will need to be oxidized to generate ATP. These sources include fatty acids, lactic acid, amino acids, ketone bodies, and muscle glycogen. The net results of the influence of increased glucagon, cortisol, and ANS stimulation during the post-absorptive state include the production of glucose from the breakdown of glycogen, proteins, and fats, and the oxidation of other fuel sources.